Hi everyone, Dr. Sadaf Qureshi here, and today we are going to talk about cell cycle. Let's start our topic with introduction. Cell cycle is fundamental process that allows cells to grow and divide, ensuring the continuity of life. It consists of several tightly regulated phases and checkpoints that maintain cellular integrity and function. Cell cycle is composed of two major stages. The first one is interphase, where the cell prepares itself for cell division. It is major part of cell cycle and includes G1 phase, S phase, and G2 phase. The other stage of cell cycle is M phase that includes mitosis and cytokinesis. Mitosis is nuclear division and cytokinesis is cytoplasmic division. Mitosis is further divided into prophase, prometaphase, metaphase, anaphase, and telophase. These stages ensure the genetic material and cellular components are accurately replicated and divided between two daughter cells by having different checkpoints at the end of each stage. They are highly coordinated processes regulated by molecular signaling pathways. Let's start our discussion with interphase, and the first stage of interphase is G1. G1 phase is also called as gap 1 phase. It is period of intense growth and metabolic activity. The processes that happen during G1 phase are the cell grow in size, it synthesizes RNA, protein and organelles and ensure it has sufficient resources for DNA replication. Cellular activities like metabolism are at their peak at this stage. The checkpoints there are the G1 checkpoints that are also called as restriction point that evaluates external signals like growth factors, intracellular condition and DNA integrity before allowing progression to S phase. The next phase in interphase we have is S phase, that is also called as synthesis phase. There DNA replication takes place producing identical sister chromatids. Processes that happen in S phase are each chromosome duplicates into two chromatids connected at the centromere. This is crucial for accurate segregation during mitosis. During this phase, histone proteins are also synthesized to package newly replicated DNA into chromatin. Centrosomes are also duplicated there. The checkpoint there is S checkpoint, and there we check for DNA replication errors, ensuring genomic stability. The next phase in interphase is G2 phase, that is also called as gap 2 phase. The purpose here is to undergo further growth and preparation for mitosis. The process included in there are synthesis of microtubules and other proteins necessary for chromosome segregation, and DNA is carefully checked for damage or incomplete replication, and necessary repairs are made in this phase. Checkpoint there is G2 checkpoint that verify whether the DNA is error-free and the cell is ready to enter mitosis. Now we have the next major stage of cell cycle, that is M phase, that is also called as mitosis or cell division. It ensures equal distribution of chromosome into daughter cells. It has several stages including prophase, prometaphase, metaphase, anaphase, telophase, and at the end there happen is cytokinesis. First we will talk about prophase, that here chromatin, that is uncoiled DNA there, condensed into visible chromosome, each consisting of two sister chromatids connected at a centromere. Then the nuclear envelope disintegrates, allowing spindle fibers to interact with chromosomes. Centrosome moves to opposite poles of the cell and begin forming the spindle apparatus. Then the next phase in M phase is prometaphase. The nuclear envelope completely disintegrates here, allowing spindle microtubules to interact with chromosomes. Kinetochore protein form at the centromeres of chromosomes, serving as attachment point for spindle fibers. Then microtubule extend and attach to the kinetochore where other overlap at the cell center and stabilize the spindle. The next phase in M phase is metaphase. Chromosome align at the central metaphase plate, that is also called as equatorial plate in this stage. Each chromosome's kinetochore is attached to spindle fiber originating from the opposite pole. The precise alignment ensures that the sister chromatids will be evenly distributed to each daughter cells. Then the next phase in M phase is anaphase. Sister chromatids are separated as the spindle fiber attached to kinetochores shorten. pulling chromatids towards opposite pole. Non-kinetochore microtubule elongate, pushing the poles apart and elongating the cell. By the end of anaphase, each pole of the cell has an identical set of chromosomes. This ensures each daughter cell receives an equal set of genetic material. The next phase in M phase is telophase. Chromosomes there decondense back into chromatin, becoming less visible under a microscope. Nuclear envelope reform around the two sets of chromosomes, creating into distinct nuclei. The nucleolus reappears in each nucleus, signaling and the end of nuclear division. The mitotic spindle disassembles as its job is completed now. And the next phase, the end phase of M phase, is cytokinesis. What happens there? The cytoplasm divides, creating two daughter cells. In animal cells, a cleavage furrow forms caused by a contractile ring of actin and myosin filaments. The contractile ring tightens, pinching the cell membrane inwards until the cell is split into two daughter cells. In plant cells, there is cell plate formed at the center of cell due to fusion of vesicles derived from the Golgi apparatus. The cell plate expands outward until it fuses with the existing cell wall, dividing the cell into two daughter cells. Now let's talk about the regulation of cell cycle. The cell cycle is tightly regulated by checkpoints and specific proteins like cyclin and cyclin-dependent kinases. Checkpoints in cell cycle include G1 checkpoint that ensure the cell is large enough and environment is favorable for division. The other checkpoint is G2 checkpoint that confirms DNA replication is complete and error-free. The other next checkpoint is M checkpoint that ensures chromosomes are correctly attached to spindle fibers before segregation. These are three important checkpoints for cell cycle regulation. So this was all about cell cycle. Thank you so much for watching. If you like our video, don't forget to like, share, subscribe and comment. Thank you for watching.